Let me start with a paradox. Why would evolution keep genetic variants that can make us seriously ill? You often think evolution optimizes for health, but it doesn't. It optimizes for survival in a specific environment. And sometimes what helped us to survive in the past make us really ill today. We encountered a striking example of this during the COVID pandemic. At that time, researchers worldwide were asking a simple question. Why do some people develop severe disease while others don't? One of the strongest genetic signals pointed to a variant called RS1138-5942. This variant sits in the region regulating the gene LZTFL1, and it carries an unexpected origin. It comes from Neanderthals. So COVID didn't just reveal a disease mechanism, it revealed a piece of our evolutionary history still active today. Now, what does this gene actually do? LZTFL1 controls something fundamental, the behavior of epithelial cells. These cells line your airways. They are first line of defense. They clear the pathogen using cilia, maintain a physical barrier, and coordinate immune response. But most importantly, they make decisions. Should they stay stable and productive or switch to inflammation and repair? This decision is controlled by regulatory signals. Two of them are especially important. First, IRF3. This is your emergency signal, activated immediately after virus is detected. Second, VDR, vitamin D receptor. This is your balancing signal. It prevents overreaction. Now, here's where our analysis began. We looked at the DNA region around this variant and asked a simple question. Which transcription factor binding sites can be found there and can be altered by this mutation? We started with JASPAR, which is a widely used resource, and we found something, but actually not everything. JASPAR detects the VDR-related binding site, but it missed actually a crucial component. Then we applied TRANSFAC, and suddenly a different picture emerged. We identified an additional binding site for IRF3. Now this is crucial, because IRF3 is not just any factor, it's essential to antiviral defense. So now this region is not just regulatory DNA, it's a decision node, a place where antiviral signal and regulatory signals integrated. This is the level of biology where outcomes are actually determined. Now let's connect this back to the COVID. SARS-CoV-2 has a special property. It suppresses IRF3. So the early antiviral response is actually weakened. Now combine this with the genetic variant. The regulatory system is already altered. The integration of IRF3 and VDR signal is distorted. So instead of coordinated response, you got a mismatch. Too little control earlier, too much reaction later. And this directly affects LZTFL1, which means the epithelial cells lose stability. Cilia functions is impaired. The barrier weakened. Now the virus persists longer, more damage accumulates, more signals are released, and the system crosses a threshold and entered into so-called cytokine storm. This is not just an inflammation, it's actually a loss of control, a network that amplifies itself through the multiple positive feedback loops. And if it continues, it can actually progress to sepsis, which is really a bad condition. But here is the key insight. COVID didn't just create this mechanism, it revealed it, because this genetic configuration is much older. In ancient environments with constant exposure to many different pathogens, a more reactive epithelial system could be probably beneficial, faster response, stronger activation, better survival. And this is likely what this Neanderthal-derived variant provided. But biology is always a trade-off. The same system that reacts quickly can also lose control. In a modern condition with pathogens that interfere with early signaling, this system becomes really unstable. So what we see is not just a disease mechanism, it's an evolutionary mismatch, an ancient regulatory solution meeting a modern biological challenge. And this brings us to a deeper conclusion. Disease is not just about genes, it's about regulatory logic, about how signals are integrated. And if you miss part of that logic, you miss the whole mechanism. In this case, if you rely only on a simplified motif libraries, you may miss a key regulator like IRF3, and then the biology remains incomplete. But once you see the regulatory code, everything connects, from Neanderthals to transcription factors, to epithelial cells, to systemic disease. One continuous system. I'm Alexander Kell, and if you want to understand disease, you must understand how genes are controlled. You can tell us about your problem by filling out the form. The link is below the video, and we'll try to explain how we can help. If your issue is more complex, simply tick the box in the form, and we'll arrange a Zoom call and talk personally. Stay tuned, and goodbye.